Hello, in this video we will understand about the vitamin B7. It is also known as biotin. So it is vitamin as a micronutrient requirement in our body. So what is the function? In this video we will understand about that. So the water soluble is an essential cofactor. It is to enzyme in intermediary metabolism and a key regulator of gene expression. So the enzymatically activity due to this vitamin is possible because it is a cofactor. So it will help to the enzyme stimulation and working well. The regulation of gene expression is also possible due to the vitamin B7. The deficiency is less possible, remember. So this is the product, the 10 source of vitamin B7. You can see the strawberry, beef, liver, chicken, liver and milk, salmon, egg, broccoli, vegetable, avocado, sweet potato and sunflower seeds, which that contain a range of the vitamin B7. So it in this way we can consume easily. The 30 microgram per day is the biotin is our requirement. In this the holocarboxylase synthetase enzyme, when this biotin enter into the cell, will lead to activation through holocarboxylase synthetase enzyme to produce biotinyl AMP. This biotinyl AMP, the holocarboxylase enzyme again work, will lead to soluble guanylate cyclase production. This soluble guanylate cyclase will lead to GTP conversion into the cyclic GMP. The cyclic guanosine monophosphate and pyrophosphate will be released. In this, the protein kinase G activation will lead to phosphorylated proteins occur in the DNA in the histone protein, the tail of the histone as a linker protein. In this way, the histone protein contain H2A, H2B, H4 and H3. In this way will lead to activation of DNA, certain gene of activation or silencing will lead to messenger RNA production, and the specific proteins will be produced like required protein. That required protein can be during the exercise, after the exercise, the actin and myosin synthesis requirement. So in this way, the more synthesis of that protein. Required biotin for biological activity is the acetyl CoA carboxylase 1 and 2, as well as pyruvate CoA carboxylase, propionyl CoA carboxylase, and the methyl crotonyl CoA carboxylase. This all enzyme required the biotin. So the biotin recycling, if we will discuss, so that it can be recycled. The diet and microbiota produce the biotin. This biotin in the presence of the apocarboxylase and the holocarboxylase synthetase enzyme to produce the holocarboxylase. In this way, the holocarboxylase synthetase enzyme produce, and this biotin linked oligopeptide formation will occur. In this way, the biocytin will lead to biotinidase enzyme lead to release the lysine and again biotin formation. Anyhow, this let's understand about the acetyl CoA carboxylase activity, the function of the biotin. For example, liver produce the low density lipoprotein and high density lipoprotein and glucose. When we will eat glucose, so the hyperglycemia will occur, and in this way the fatty acid will be rise whenever you will eat the fat. Insulin bind with the cell will lead to GLUT4 channel open to influx of the glucose, while the fatty acid can be directly moved because it is lipid soluble. Anyhow, remember the glucose is the fat insoluble, so in this way the water soluble required channel. A fatty acid when enter will convert into the acetyl CoA, and the glucose through glycolysis to produce pyruvate will enter into the pyruvate channel, transported channel, and the beta oxidation will occur in the mitochondria of the acyl CoA production from fatty acid. In this way, the acetyl CoA formation in the mitochondria will lead to Krebs cycle activation, and in this way the 2 carbon containing acetyl CoA will bind with the oxaloacetate to the citrate formation, 6 carbon containing molecule. In this way, citrate can be moved out through citrate transporter channel. In this way, the oxaloacetate, whenever released, will lead to again two molecule producing in the cytoplasm. And this acetyl CoA, which that will convert in the presence of the acetyl CoA carboxylase 1 and 2 enzyme, and this will lead to help as a cofactor biotin will produce the malonyl CoA. This malonyl CoA will convert into the palmitate and acetyl CoA again. This acetyl CoA again will convert in the mitochondria or outside, and whenever it will be going to the mitochondria, the lipogenesis can be occur. So in this, the fatty acid will again convert via acetyl CoA reverse reaction, and also the gluconeogenesis can be possible. So in this, the metabolic pathway is important through vitamin B7 because it is cofactor via several enzymes. This is the protein amino acid isoleucine, leucine, avaline. This isoleucine convert with the reaction to produce the propionyl CoA, and the leucine produce 3-methyl crotonyl CoA, and the valine produce the propionyl CoA. These all intermediary molecule in the presence of like the propionyl CoA, in the presence of the propionyl CoA carboxylase with the biotin addition as a cofactor to trigger to produce the succinyl CoA, and for the 3-methyl crotonyl CoA to the 3-methyl crotonyl CoA carboxylase enzyme with the cofactor to bind to produce R succinyl CoA and acetyl CoA. This is the important for the chemical reaction and produce more ATP. That is why it is the metabolic vitamin which that will lead to increase the metabolism. Like it will convert this ATP from the protein, so the proteolysis can be possible due to the cofactor vitamin B7. This cholesterol also convert due to the propionyl CoA carboxylase enzyme with a cofactor biotin to produce the propionyl CoA. This propionyl CoA will convert into the D-malonyl CoA to convert the L-malonyl CoA to produce succinyl CoA. This is same mechanism for the Krebs cycle. It is used for the Krebs cycle to produce ATP, and this ATP and NADH and FADH2 is used for the electron transport chain to produce more ATP. That is why it is the ATP production vitamin B7. The biotin containing propionyl CoA carboxylase in metabolism of methionine and threonine we will discuss. It is also the methionine will convert into the S-adenylate methionine, and the S-adenylate homocysteine conversion will lead to homocysteine formation, and the cystathioine produced will lead to cysteine separate. This is the amino acid, while the alpha ketoglutarate will produce again the succinyl CoA in the presence of the propionyl CoA carboxylase enzyme, while on the other hand the threonine also convert into the alpha ketoglutarate. This is the all metabolic activities can be possible due to the vitamin B7 as a biotin. And old chain fatty acid also convert into the acetyl CoA released to propionyl CoA. The conclusion is that the vitamin B7 is important for metabolism of glucose, protein and fats, for production of ATP. That is why the normal metabolism required this vitamin B7, and it will help to produce ATP via a cofactor binding with a specific enzyme. So the risk factor for biotin deficiency is smoking, alcoholism, a white egg prolonged consumption, cirrhosis in the liver, anticonvulsant medication, and deficiency and sign and symptom we also understand. But remember, this is the anticonvulsant medication is also cause this risk factor for biotin deficiency. Let's understand about the deficiency and sign and symptom. It is alopecia. the alopecia, neuropathy due to the nervous system dysfunction. Due to this enzyme is required the energy, produce the energy, but the dysfunction of the energy production will lead to decrease the metabolism and cause the neuropathy. Rashes in body, depression and the hallucination, numbness, ataxia, seizure, metabolic disorders, and susceptible to viral and fungal disease because our immune system required this all activity. So this activity inhibition, this metabolic reaction inhibition will lead to inhibit the function of the immune system well. So in this way it will lead to cause more susceptibility. While the nervous system also required this metabolic pathway for the oxidation of fat if not possible, so the deposition of fat will increase lead to cause also obesity, as well as the deposition of the fat in the brain will lead to cause many problems. Thanks for watching.